Hello, everyone, and welcome to another webinar from the Science and Technology Committee from AMM. Today, topic going to be manufacturing processes of different mRNA modalities. We have three excellent speakers that they're going to dive into self-amplified manufacturing process, circular mRNA, and continuous manufacturing. The agenda today will be a brief introduction to AMM, and also continuing the AMM Science and Technology Committee webinar series. After our three speakers, we'll have a roundtable discussion with Q&A. Please hold all your questions, submit them through the question portal, and we will address them during the Q&A session. Next. Clay, you want to take that on?
Go ahead, Colin.
AMM is the Alliance for mRNA Medicine. It's a consortium that started a couple of years ago to advance mRNA therapies and be an advocate for the advancement of mRNA for use in medicine. And we strive to help patients and public health and society to benefit from this new modality. The mission is to propel the future of mRNA medicine, and AMM is a consortium that include industry leaders, innovators, scientists, academics, and nonprofits that come together to look, to advance mRNA therapies. Next. What does AM do? We try to educate and demystify the technology by explaining how mRNA and next generation encoding RNA works. We engage with regulators, policymakers, and other stakeholders in North America, Europe, and Asia Pacific. And that's mainly to advocate policies and encourage innovation and help shape the regulatory standards, support manufacturing, and promote access of mRNA medicine to patients. AMM is developing the most comprehensive source of reliable mRNA sector data. So we actually collect all that data and make it public so people know what's going on. And AMM convenes the community and amplify the work of scientists and organizations seeking to improve health through mRNA and next generation encoding RNA. And next slide. So this year we have our second annual meeting, and this year that meeting is going to be in Coronado, San Diego, September 16 to between September 16 and 18, 2025. We look forward it's open to public, and we look for a lot of people to attend and gain a lot of knowledge about the mRNA field. Next slide. AMM Science and Technology Committee webinar series. This is the third, actually the fourth one that we're putting out, and this is going to continue all the way to December on a monthly basis. So the committee is to advance emerging scientific issues and work with mRNA community to support, develop, and implement cross-cutting standards and workforce training, and basically trying to share with the scientific community the new advances on what's going on in terms of new advances in mRNA. The goal of the webinar is to discuss critical scientific topics, and we're trying to put one every 4 to 6 weeks. Coming up is we have the AMM Innovation Forum that's happening on June 16 in Massachusetts. It's building on this webinar series. The meeting will provide in-person forum, and also you can register and attend virtually. It's a one-day forum packed with speakers that will dive into the scientific application of mRNA from the medicine standpoint, and also the raw material processes and regulatory requirement. Next slide, please. The next Science and Technology webinar, it will be on June 24, and this will dive into the characterization of mRNA. Please register today if you're interested. And the next series, the continuing series will be going over the quality attributes for mRNA, and then from there we'll switch to delivery, going over delivery strategies, characterization of the drug product, and the quality attributes of the drug product, and we will finish the series with the CMC requirement for mRNA therapies. Next.
So today we have a great panel for our discussion. It'll be moderated by Khalid Yamut, who is the co-chair of our Science and Technology Committee. Kicking off our presentations will be Andrew Gill, who is the Chief Development Officer of Replicate Bioscience, followed by Andrew Varley, who is the Director of the University of British Columbia, and then Zoltan Kis, who is a Senior Lecturer of the School of Chemical Materials and Biological Engineering at the University of Sheffield. So with that, I will turn it over to Andy to get us started.
Thanks, Ashley. So next slide. And next slide. Okay, so today I'm going to be talking about how to produce self-amplifying RNA using an in vitro transcription reaction. I'm not going to give you a secret recipe. I'm kind of going to guide you as to how and why you might go about selecting certain parameters, where information in the public domain exists to inform you and make the right choices. Self-amplifying RNA or self-replicating RNA is an mRNA. It has a 5' cap, a poly-A tail, and it encodes genetic material. The difference is it encodes the non-structural proteins for an alpha virus, allowing it to self-amplify or replicate in a cell. This means it's much larger, and you need to take into account that fact during manufacturing, and we'll come on to why in future slides. So next slide. Mechanistically, this is what it looks like. So down the bottom, we have a messenger RNA. You deliver it to a cell using a lipid nanoparticle. It engages the ribosome and produces your therapeutic protein. In most cases right now, that's an antibody, but it could an antigen. It could be, a therapeutic antibody in the future, or some other therapeutic protein. The self-amplifying RNA gets delivered to the cell and then ends up in a sphereole, this membrane-privileged Breb, and it goes through its replication cycle, makes much larger quantities of endogenous mRNA encoding the antigen or therapeutic protein. So you get higher levels of output and greater durability. So next slide. But these molecules are much more complicated to make. Certainly, I've been in this field since 2008, and back then there wasn't much information available about the enzymatic production of RNA, and a lot of work had to happen. Since then, there's a lot more information available, and hopefully I can guide you to where that information is. But this is a schematic describing that enzymatic transcription reaction. It's cell-free, and for the pharmaceutical industry, that's a huge deal. We no longer have to purify away our drug substance from the contaminants that come from cell culture. We need a polymerase, T7 polymerase. That enzyme reads the code in a DNA template in a sequence-dependent manner. The template needs to have a promoter region to allow the T7 to bind. It unwinds the template and then reads down the transcript. It incorporates nucleotides. These are supplied as nucleotide triphosphates in the reaction mixture. You need to add a number of reagents in order to optimize this reaction. RNase is one of the contaminants that can be introduced. Humans shed RNase 7 from the skin. We breathe it out. If that enzyme ends up in your enzymatic reaction, you will degrade the material as it's being made. So an RNase inhibitor is added. One of the byproducts from the reaction is pyrophosphate. And so an enzyme, inorganic pyrophosphatase, is added, removing that contaminant as it appears, because it will limit the yield of the reaction. Magnesium is added as a counterion to help incorporation of the NTPs. And then you have reagents like spermidine that promotes the RNA stability, antioxidant to stop the enzymes oxidizing DTT, and then a buffer to control pH. In this example, we're using enzymatic capping. So there's a capping enzyme added in multiple components. You could use co-transcriptional capping, and we'll come on to that in later slides. The DNA is removed using an enzyme as well, a DNase, and we'll go through the sequence of events in a schematic later. So next slide. So this is what was available in 2008. There were kits available from people like New England Biolabs, and this is a kind of a process flow of that kit. You take your DNA template, you add enzymes, you incubate, you then add the DNase to digest the DNA template, and you can see the times and temperatures listed there. You then precipitate out your RNA, you wash with ethanol, you clean it up, resuspend it, and then you're ready to go through the capping reaction. You add your capping reagents, again, go through lithium chloride precipitation, washing with ethanol, and resuspension, and you have your drug product. Obviously, this is a multi-step process. It's not ideal for producing RNA at a commercial scale. So next slide. Just to note that there aren't many publications describing the large-scale manufacture of self-amplifying RNA, and this is probably due to the stability profile relative to short RNA. And I'll point you to this literature where it goes through, stability profiles of RNA by length at the bottom there. Next slide. So if we look at the major degradation pathways of RNA, the first one is that enzymatic degradation RNase. So you need to have a containment strategy for producing RNA. Avoid the introduction of RNase enzymes, either in the reagents going in or contaminations coming from the environment. That containment is straightforward and common in many industrial closed-loop manufacturing processes. So it's not actually a major issue. The major issue for self-amplifying RNA is hydrolysis of the phosphate backbone. And this is time, temperature, RNA length, pH, and buffer species dependent. And so here's a little bit of literature available. And so in the graph, on the left-hand side, you can see a couple, two different pHs, pH 6 and 7, and some buffer species. And you can see normalized to percent RNA integrity, so the amount of full-length RNA present after incubation at elevated temperature, 60 degrees for 3 hours. And what you can see is it's definitely pH and buffer species dependent. Just to note, the tris buffer there at pH 7 is very similar to the buffer used in the one of the commercial COVID vaccines. Next slide. So here, just depicting graphs showing length-dependent hydrolysis at different temperatures. So you've got samples 1 to 5 ranging from 500 nucleotides close to 6,000 nucleotides, and we're going from 40, 50 to 60 degrees C and measuring time-dependent integrity. And it's quite clear that the longer the RNA, the faster the hydrolysis. So when you're developing your process for a self-amplifying RNA, reducing time and temperature and picking the correct buffer pHs are essential. Next slide. This is just a schematic of what a commercial process should look like. You have a cell-free bioreactor where you conduct your in vitro transcription reaction. You're able to control temperature, stirring of the material, and pH. Then you need to go through a purification process. After the RNA is purified, you're going to then need to dialyze and concentrate the material using tangential flow filtration. And finally, a bioburden reduction through a 0.2 micron filter. The composition of that IVT reaction is well defined, and so you can control the residual material coming from it. The yields are usually quite high for self-amplifying RNA in the 5 to 8 milligram per mil range. It's a one-day process, and it's antigen or gene of interest in coding independent, although for each construct, minor optimization will need to happen to both reduce residual contaminants such as double-stranded RNA and optimize the yields of the process. And certainly this process is used for all types of RNA. Today I'm talking about self-amplifying, and others will talk about other types of construct. Next slide. This is just a quick note on the different ways of capping your mRNA. This is stolen from NEB. The citation is down there on the bottom. On the left-hand side, you can see post-transcriptional capping using an enzyme. So you take your DNA template, you make the RNA, and then on the 5' end, where there's a triphosphate, you add enzymes. NEB now offers 2 types. Historically, the vaccinia capping enzyme, which is a multi-component reagent system that can produce either cap0 or cap1 is added. I'll later on talk about the Faustus enzyme and how that might help optimization for production of self-amplifying RNA. So using this process, you're able to put a cap1 efficiently onto the mRNA. On the right-hand side, you can see co-transcriptional capping. The advantage to this is it's not a multi-step process. The cap is incorporated at the beginning of the reaction, and then the in vitro transcription builds out the construct from that cap. Historically, yields have been low for longer RNA using this technology, although more optimization has happened at places like TriLink and others, and yields have increased to be equivalent to the enzymatic capping process. Next slide. This is a paper published in 2024. If you're interested in understanding the multifactorial optimization of an in vitro transcription reaction, specifically around optimizing longer RNA, this is a great place to go. And it highlights that magnesium, time and temperature dependence are those major contributors to optimization of the IVT reaction. Next slide. This is what a process flow looks like. So you start with the DNA, you linearize it. I'd suggest you take a good hard look at the purity of that template. It's critical for production of high quality material, independent of what RNA you're making. And certainly pause there, characterize your template before it goes into the IVT reaction. Once in the IVT reaction, this is for enzymatic capping. The IVT is performed, you digest the template, then you add the enzyme reagents and go through chromatography, dialysis, and pH adjustment of concentration and sterile filtration. Certainly, historically, that multi-step process was not ideal. Now we're starting to see for the enzymatic capping processes and multi-step in the same bioreactor process is being adapted. And so the process flow looks very similar to co-transcriptional reactions, and you don't have to go through intermediate purification. Next step. Now, there are many types of RNA and many different types of applications, ranging from vaccines to cell therapy, genome editing, and therapeutic antibody expression. And the types of RNA you use will be critical. Not all will be ideal. And whether you use a base modification or not will probably depend on whether you're in the vaccine space or not. Next slide. This is just a reminder, most of the self-amplifying RNA systems that we saw go through into COVID clinical trials did not contain a base modification. Moderna and Pfizer-BioNTech used N-methyl pseudouridine. There's actually one example that did for self-amplifying RNA, but the majority did not. Next slide. This is just a snapshot for those interested of some of the major clinical trials in self-amplifying RNA in the COVID-19 setting. The one down the bottom, VLP Therapeutics, is the one that used 5-methyl cytidine as a base modification. They were having high levels of reactogenicity, and they introduced it to try and dampen down reactogenicity. It certainly worked. It didn't go on to later stage development. The one that won through was the Arcturus, and they received approval in Japan very recently and showed great data as a booster vaccine, showing greater induction of neutralizing antibodies and greater durability. And again, it's an unmodified system. Next slide. This is just a publication in 2020 summarizing the 143 different base modifications. If you're interested, down on the figure 3, you'll see those 22 that are included in mRNA. And obviously methyl pseudouridine or pseudouridine is the major base modification used, but there are several others that can be used. Whether it can be incorporated into an mRNA will be dependent on the T7 polymerase. And so many of these won't incorporate in efficiently. There are about 4 or 5 that will, and certainly for self-amplifying RNA. Next slide. M5 cytidine was one of the key components. Here's a recent publication from a group in Boston showing that substitution can be incorporated efficiently, and they claim reduction in immune stimulation of the molecule. So if you're interested in gene therapy, this might be a good place to take a look. Next slide. So with self-amplifying RNA, highlighted that, hydrolysis is time, temperature, pH dependent. It's also magnesium ion concentration dependent. And so as you optimize your IVT reactions, it's probably a good idea to have some analytical assays that will tell you what's going on in that reaction. And certainly historically at Replicate and other companies, we focused on measuring integrity using capillary gel electrophoresis and using an in vitro cell-based potency assay to show where optimal conditions exist based on the ability of the RNA to self-amplify and express the encoded protein. Next slide. Historically, agarose gels were used to characterize longer RNA. You can see an agarose gel here. Just looking, we have a molecular weight ladder in lane 1. Lane 2 is a reference standard. We have a main band with a smear below, and then again, the lane 3 molecular weight marker and our test sample. And comparing the 2 lanes, it's difficult to quantify what's going on between the 2 samples, between the test and the standard. So next slide. So we at Replica worked with SciEx to develop capillary electrophoresis systems to measure these longer RNA. This is an example of a 9.4 kb RNA. Up the top, you have electropherogram with the RNA ladder going out to 9 kb and a reference RNA versus a test RNA. You are able to integrate that electropherogram and come up with a percent corrected area, the reference standard being 53%, the test sample 42%. So this was a big step forward in characterization of self-amplifying RNA. Next slide. And so here we're just looking at a stability of a longer RNA. We're degrading it in a time and temperature dependent manner. You can see on the agarose gel, the main band is disappearing and the smear is increasing. In the middle, you have densitometry on the agarose gel. And then on the right-hand side, those samples run on the CE method. And you can see quantification of the percent main peak, either numerically or on the graph down the bottom. So we see a decrease in integrity as we increase the harshness of that degradation of that self-amplifying RNA. Next slide. So In a cell-based potency assay, we can measure the ability of the RNA to replicate in the cells. You can measure that double-stranded intermediate in cells using the J2 antibody. That's what it was originally developed for. And so using a reference standard, you can generate a potency curve shown here on the left, different amounts of RNA electroporated in. If you then take those test samples that were degraded and electroporate them into RNA, you can measure the percent potency relative to the standard. So you can see that decreasing over time down to 0. And then you can correlate potency versus percent main peak by capillary electrophoresis, and the R-squared value is 0.98. So there's a direct correlation between CE percent integrity and the ability of the RNA to replicate. And so we're able to probe and test that IVT reaction at multiple points along the way to determine where and how to optimize the production of highly potent, intact, and full-length RNA. So next slide. So IVT reactions and the components available haven't stood still. Historically, batch production is common across the mRNA field. During the pandemic, we started to see self-amplifying RNA. It took some time for those IVT reactions to be developed. Now many CDMOs can make longer RNA. If you're interested, the way forward is to probably go under a material transfer agreement and get them to make a small sample for you. But also, the IVT components have changed and been optimized. So we have now synthetic DNA templates that don't require E. coli for production. We have new enzymes, new cap analogs, better purification. It's been characterized extensively in the literature how to reduce residual contaminants such as double-stranded RNA by optimizing the IVT conditions and or removing them through downstream purification. We've seen automation of the IVT process by people like Quantum, so robots that can now produce material and reduce the operator error. We're seeing continuous flow processes from people like Centillion and Nature's Toolbox, where RNA is produced in a very short time period and harvested, but in a continuous flow. And then continuous production is being characterized where the IVT reaction is run over 24 hours and continuous harvesting takes place. Next slide. So there's a lot happening and it's all beneficial. This is a citation just showing one of those new enzymes that is able to reduce contaminating double-stranded RNA during production. Just one word of caution, take a look at your yields. They're not optimal for self-amplifying RNA. Next slide. So this is just a snapshot really of what's going on. This is something I pulled from New England Biolabs, their Faustus enzyme. It's a single component enzyme now, much lower costs. And we're just looking at percent cap incorporation over time, over different temperature ranges relative to the old vaccinia. And what you can see is you can reduce the temperature of the IVT and still see good incorporation of cap down to 25 degrees. That may have an advantage for these longer RNAs that are more temperature dependent and time dependent in the IVT reaction. Next slide. Just a little note on cap analogs. Rather than using enzymes, during COVID, we saw a switch from ARCA, this anti-reverse cap analog, to reagents like CleanCap. And next slide. Now you can get capping reagents for self-amplifying RNA. They require an AU start rather than an AG start. Those are now available from TriLink under GMP production conditions, so could be used for your IVT reactions now. Certainly not available during COVID. Next slide. So certainly lots of choices around enzymes. So with that, I'll say thank you and we'll take questions at the end. Going to hand you back to Ashley.
Thanks, Andy. Up next, we have Andrew Varley.
Great, thanks. So just a quick little correction I want to make right away is I'm not the director of UBC, as exciting as that would be. I'm the director of the RNA and Formulation Core at UBC. And yeah, I'll be talking about the circular RNA manufacturing challenges and everything. And very similar to Andy, you know, we don't have a secret recipe we can share or anything, but we can follow along with what Andy was talking about. And as he's covered a lot of the fundamental IVT challenges, I'll focus more on what's really unique to circular RNA. So next slide, please. So circular RNA itself, you know, as it sounds, is a circular product. It lacks those 5' and 3' ends. And this imparts some really interesting properties to it. The main being that when messenger RNA or self-amplifying RNA is inside cells, the main degradation pathway relies on degradation of that 3' poly A tail. Once that gets shortened, then it gets decapped from the 5' end and then rapid degradation of that. So clearly, circular RNA, once it's inside cells, is a lot more resistant to these kind of pathways, which is really exciting. There are some sequence or structure-based mechanisms that do degrade circular RNA in cells, and it is also susceptible to, next slide, the autohydrolysis that occurs. So as a quick kind of highlight, this isn't actual data, but just a representative figure. On the top right there, we can see mRNA, once it gets in cells, usually has a really strong peak expression, but doesn't last very long. Self-amplifying RNA lasts for a very long time, which is kind of a misnomer. It replicates, which allows that expression to happen for a very long time. Whereas circular RNA actually does last a bit longer. The RNA itself is fairly stable. And so we get somewhere of some sort of intermediate type expression. Next slide, please. And as Andy was mentioning, not too long ago, we saw the first marketed approval of an siRNA modality. This is kind of an overview of what we've seen in the field so far. siRNA, it's not translatable, but was one of the first hallmarks of success in the RNA field. In 2020, we saw the COVID vaccines, and just more recently in 2024, we saw the mRNA RSV vaccine. So when are we going to see a circular RNA modality? It's quite an interesting question, and I think something that we'll see fairly soon. And I'll touch base on where the status of the field is in a little bit. Next slide. Now, one of the most interesting things about circular RNA is how do we synthesize at scale? There's ligation approaches that typically don't work too well once you get above about 200 nucleotides. In contrast, we have autocatalytic mechanisms, such as through group 1 or group 2 introns. And that's what we see here on the left two figures. In 2018, Alexander Wesselhoff and his group with Dan Anderson came up with this really clever way of basically reworking how splicing occurs to allow circularization autocatalytically. And it uses, like I said, these introns. But really importantly to this process, you see in the bottom left, there's those little white segments in the final product. And that is essentially a scar. Those are leftover regions of those introns. There has been some engineering for these group 1 introns, as we see in the middle, that can essentially form a circular product without a scar. And not highlighted here, there's group 2 introns that can also do this as well. Over on the right, there's this new approach in which it's essentially a ribozyme-mediated ligation. And this is a really exciting different process. They showed very high efficiency, as you can see in orange there, close to 95%, whereas an enhanced or highly efficient intron-based system that they compared to anyway was about 86%. And they use this as an antiviral type approach. Next slide, please. And so one of the main things that we have to think about with circular RNA is the fact that it doesn't have those 5' or 3' ends. And so lacking a 5' end, it can't use canonical translation initiation that's 5' cap dependent. And so we instead rely on something called an iris, an internal ribosome entry site. And there's lots of different things at play when you're actually doing this synthesis and design. If we first take a look at the top right there with ribozyme selection, those are your group 1, group 2 introns or some sort of ligation approach. But you also have to include homology regions and spacers to make sure this happens efficiently, because you need to make sure that those actually come together effectively. The iris, generally speaking, we see around 700 bases for a potent iris. And so you can play around with lots of different irises, but there's some things to consider, such as the gene of interest could potentially interfere with your iris. So the iris is highly dependent on structural elements. If your gene of interest has some interaction with those structural elements, you can actually get reduced potency for that reason. As well, a lot of irises are from viral origin, and so there's a potential for some immunogenicity from there as well. The one thing I didn't quite highlight yet as well is those ribozymes are also susceptible to sequence-based interactions with either the iris or the gene of interest. So there's a lot of structural important aspects to circular RNA that have to be considered, and things like RNA fold and stuff can give you some hints to what's going on there, but there's not really good modeling for these kind of things yet. And lastly, poly-A tracks and other untranslated regions can also make a big change on how well your circular RNA is going to work. Next slide, please. So highlighting a little bit more of the iris itself is some of these different technologies here, in which on the left we see from Howard Chang's group. They actually took that iris and did some engineering, added some aptamers to try to improve the ribosome recruitment. And on the right, this is a really nice fundamental study, really showing the types of elements inside an iris that make it work really well. Next slide. Now, I had mentioned that you can't use a 5' cap for circular RNA, and canonically that makes sense. However, there's been some really interesting advancements in the field recently, 2024 and as recent as 2025, where they're finding creative ways to reincorporate the cap essentially. And on the left, what we see is an approach where they synthesized an oligo with a branched region that allows cap addition, which is really, really exciting. It is a very big challenge for a synthesis and purification, well, primarily a synthesis approach, because like I mentioned before, once you get large circular RNAs, the ligation method doesn't really work that well. So really creative. One thing I do want to highlight here, and we see these really big differences in the total flux and the luminescence in that study, but they did use completely different synthetic approaches here. So like I said, with the cap, they used ligation approach, but with the iris mediated expression, they used the typical group 1 intron. And there's a lot of questions about purification when we talk about doing the autocatalytic methods. And so one thing to always highlight, especially in RNA research, is that really the way that you analyze things, the way that you purify things and characterize things will make a big difference on how you actually get your results, because it's very easy in a lot of ways to not sufficiently analyze your RNA and then be able to have nice results because of it. On the right here, we see internal cap initiated translation, which is taking similar things that they did in that previous study and kind of taking a bit of a step further. They recreated a very similar approach. But then if you take a look on the right, where we have this highlighted on, this was a creative way to essentially not require this whole ligation process and just take a little oligo that hybridizes with your circular RNA that contains that cap. And so in this case, you can actually introduce a 5' cap in that region of interest and create expression. So really cool non-covalent bound cap through that process. And what I thought was really, really exciting about this paper is they also said, okay, well, if we can do this, we can also potentially just use endogenous transcripts to do the same thing. And so on the bottom, what they've done is design the circular RNA to be complementary to an endogenous mRNA, and essentially bringing endogenous mRNA caps into the right proximal region to create the translation from the circular RNA. So this is really cool, and in potential for doing some cell-specific expression based on that design. Next slide. Now, since we kind of talked a little bit about how circular RNA works, I want to really focus on all the different byproducts. And we see the same byproducts that we get from a typical IVT reaction, plus some extra ones that are introduced from circular RNA. So in the middle, we see our circular RNA, our nice target product of everything here. And to the right of that is a concatemer. So it's very possible in solution, and this is somewhat concentration dependent, whether or not a strand finds another strand beside and will actually concatemerize and create a nice long circular RNA. Functionally speaking, these should work just about as well. So they're not really a really bad thing to happen, but something that's important to characterize. And on the top, we see a lot of similar things that we get from normal IVTs. We get nicked RNA, early termination, a lot of double-stranded RNA. But the other thing that can happen is once you have your circular RNA, like I said, autohydrolysis still occurs. And so now you have linear RNA that is literally the exact same size as your circular RNA, with the only difference of one covalent bond. So this can be very challenging to purify. And one of the results of these group 1, group 2 introns is once you circularize, you have these little fragments that are left over floating in solution. And they have some degree of double-stranded RNA nature, but there are also going to be a lot of these little fragments, and those can act as templates for other double-stranded RNA. So one thing that's really important to consider here is the longer your circular RNA, the more concatemers and a lot more failed circularization events that you'll get occurring. Next slide. I'll just highlight some interesting data. We're not the first to, well, we haven't actually published these, but there are published literature that highlight these, but we're just kind of sharing some things that we've done in the past. And that's introducing multiple genes into a single circular RNA. Nothing incredibly novel here, but it's really interesting to be able to note that, you can introduce the exact same iris multiple times in a circular RNA. We've actually done it with 3 different genes, and we see pretty stoichiometric expression, which is really nice if you're trying to think about maybe producing something where like an antibody where you want fairly similar expression for like a heavy chain and a light chain. But as I mentioned, as you get bigger and bigger circular RNA, the production becomes more challenging. And then on the right, we essentially just looked at the introns as a proxy for how well we had circularization going. So in those white circles, and essentially as we increase the amount of modified nucleotides that we introduce to circular RNA, we get less and less circularization. And that's because modified nucleotides will disrupt the structures required for circularization. They'll also disrupt the structures required for an iris. So typically speaking, we don't see much modified nucleotides going into circular RNA. Next slide, please. So I'm going to touch base now a little bit on the purification. RNase R, as we see in the top right, is one of the most commonly used enzymatic approaches. And RNase R is a highly processive 3 to 5 prime exonuclease. So it's really effective at chopping up linear RNA, and it sounds kind of like the perfect solution. But the reality is there's a lot of structural elements or maybe double-stranded RNA elements that resist RNase R degradation. And so if you were just to throw RNase R at your circular RNA, it really doesn't do the job, but it gets you a lot of the way there. It does a lot of the work that you need to do. On the bottom right, we have chromatography approaches such as size exclusion chromatography, ion exchange chromatography, ion pair reverse phase, and these all have their own merits. They've been used in different degrees of success. Another one that's kind of come out more recently is cellulose chromatography, which I will discuss a bit later. And TFF is an intermediate step in a lot of these. So as soon as you start looking at scale, you can't avoid really doing TFF. But there is some potential, if you select the pore sizes properly, for TFF to remove some of these introns and start getting you part of the way there for the purification as well. And last but not least, the phosphatase, as we see on the top left. This one is a really nice and important polishing step for circular RNA because it's so hard to remove every single little bit of linear RNA, particularly as you're doing those purifications. You're going to get some nicking here and there. And those 5' phosphates that exist are highly immunogenic. And so a nice polishing step that's done on a lot of circular RNA is a final phosphatase treatment. Next slide. So talking a little bit more about RNase R in particular, we decided to take a look at a bunch of different vendors and try to find out how much endonuclease activity is found in our exonuclease preps, because any endonuclease activity is going to really reduce the amount of product that you get in the end. We also took a look at how well these purified things, but just as a quick pass here, we optimized this around vendor 2, so V2, and we analyzed things at 15, 30, 45, and 60 minutes. And what we see V3 with vendor 3 seemed to have a lot of endonuclease activity. As we can see, our yields were about half of what we were seeing for everything else. Whereas vendor 4, we got the maximum amount of yield for any of these. And in discussion with them, we know this is primarily because it went through really extensive purification. So it actually went through 3 different purification processes. And while these were exciting, they were also kind of like, okay, well, this is a lot of work to get there, so we need to figure out what's actually worth it. So next slide. Taking a look at the purification profiles from these, I'm showing here at 15 minutes and 60 minutes and the stacked on the right. Generally speaking, they achieved fairly similar purification for all these different enzymes. V3, that was the one with the low yields, was a little bit messier, and whether or not we're seeing some maybe residue from the RNAs are itself is unclear. But one thing that's really important is a really good analysis system, and we've kind of settled on HPLC, size exclusion chromatography, as a really nice first pass on understanding how well we're purifying things. And so we can see there's introns in the figure on the left. Those are removed very effectively. For the circular RNA, we seem to lose a bit of that peak. However, there's also a possibility that some of our linear RNA is in there as well. We clearly see some linear products over on the left and potentially some concatemers. So there's a lot of characterization that still needs to go into this. But one thing to mention is that we do pre-treat our RNAs, our samples in this case, with a polyadenylation step. Because like I was mentioning before, RNAs has a really hard time dealing with some structures or something like that. And so doing that polyadenylation step really improves its efficacy. Next slide. And this is, I kind of highlight this because size exclusion chromatography has come a fairly long way since its early stages for circular RNA work. And so in 2019, Alexander Wesselhoft, he did a really nice study in which he was using size exclusion chromatography. And what he discovered is that there's a lot of coelution of your nicked RNA in that main peak. So like I was mentioning in that previous slide, I think there's probably a lot of nicked RNA that we were purifying in that main peak as well. And what he found really is to get a nice pure product, you had to essentially just take the second half of that main peak. And so from a purification standpoint, he was getting really quite nice high purity, but there was a huge amount of loss in yield. So size exclusion chromatography systems really have poor resolution, but they're a really nice way to kind of get some nice data for a nice pure product. And that's what we see on the bottom. He was showing that the immune responses that we can get when there's a really good pure product are really, really quite nice. And you may notice in this figure as well that there's this circular N1 methyl pseudouridine. And this is produced from a ligation approach rather than the autocatalytic. So that's how they managed to get those. Next slide. And as Andy was mentioning, I think we might have skipped a slide there. Can we just go next? Okay, something went out. Okay, I don't know if we lost a couple slides there, but yes. So one of the things that this group was doing quite recently in 2025 was taking a very holistic approach to the approach here and taking a look at a three-step processing. First with RNase R digestion, which is commonly used. And then they used two different types of chromatography, not at the same time. So they evaluated the efficacy between the two. On the top, they used size exclusion chromatography, similar to what we saw before. And on the bottom, they used cellulose chromatography. And one thing to mention here is they used wood-derived macroporous cellulose. So it's a little bit different than this crystalline cellulose that people are often using in this, which just seemed to work quite a lot better. And then they, like I mentioned before, kind of polished it off with a phosphatase treatment. And what we see, or what they saw, I should say, is with CAR T cell engineering, this wood-derived macroporous cellulose in that figure B gave really, really nice CAR intensity results, fairly similar to just grabbing that late versus the early size exclusion chromatography peak. And they got really nice CAR intensity and cytotoxicity results from these. So this is a really exciting approach for this. Next slide. Okay, yeah, sorry, I had a mix up on my slides on this side. So one thing that Andy was mentioning with the self-amplifying RNA is how important different types of approaches on analyzing these things, such as capillary electrophoresis, really is. And truth be told, these aren't the nicest electropherograms you've ever seen, but it's also a nice way of analyzing circular RNA outside of just using size exclusion chromatography. And we're re-improving these methods right now. But as we can see, it's a nice way to kind of see what is in our solution. Next slide. So one thing that we haven't touched upon yet is the double-stranded RNA conundrum. So this is something that has been shown over and over to be really detrimental in a lot of in immunogenicity for RNA. And a quick pass, if we want to take a look at the top left there, when we were loading 200 nanograms onto a slot blot, we saw mRNA has quite a lot of double-stranded RNA. When we introduce these modifications, it dramatically reduces it. And siRNA, we're just kind of evaluating that as well. So we can see quite a lot of double-stranded RNA when we don't have modified nucleotides. On the bottom left, we took a look at cellulose purifying these, and we found as a general rule, the more double-stranded RNA that you start with, the easier it is to remove. Or there may be some kind of impact based on the fact that we're using modified mRNAs. That's something for us to tease apart. But if I take your direction up to this lithium chloride purified circular RNA that we made on the top right, slightly different concentrations than the 200 nanogram, but even down at 50 nanograms, we see a much stronger response from our J2 antibody. So when we're creating these circular RNA preps, we are getting a lot of double-stranded RNA coming out of this. And something that we started doing, next slide, is taking this approach and modifying the purification. And this is something, to be completely honest, we did not expect to work this well when we started giving this a shot. So we were really excited by these results. We were able to reduce our double-stranded RNA by over 90%, essentially just by tweaking this lithium chloride protocol and making sure that we can selectively reduce the amount of double-stranded RNA that's being picked up. And just out of interest sake, we essentially took a look at what we removed, and it was so incredibly abundant, it just made a mess of our slot plot here. So quite confidently, we're able to remove double-stranded RNA. And I had actually initially thought that this may have been from intron removal, but looking at size exclusion chromatography, that didn't seem to be the case. So we were quite interestingly selecting, I think, for long double-stranded RNA. Next slide. And one other way that we are purifying things, this is actually some quite old data that we need to revisit sometime soon as time allows, is ion exchange chromatography. And at least in this example, I will say this took weeks of optimization to get the gradient just right for this. But once we did get this really finely tuned, we were able to very confidently just extract our circular RNA. Our yields were not amazing for this, so there's still a lot of optimization going into these processes, but really excited to see how this can work without even a first initial RNase R step. So this was just by putting in completely crude circular RNA. Next slide, please. And some more work that's being done in this space, especially in the purification, just actually last month, is a really exciting paper. On the left, they did some work on optimizing the group 2 intron system by essentially introducing different homology regions, different spacers, and they actually settled on this dengue fever virus UTR. So they got really high circularization. But what I really want to highlight here is they took an approach of taking a split aptamer. So each side of that circular RNA has half of an aptamer. Once it becomes properly circularized, the aptamer is now in full conformation. And this is really cool because this is streptavidin aptamer, and this allows them to purify in a completely orthogonal approach by using affinity chromatography. And what we can see is that at least in HeLa cells, we got a very low interferon response and a good luciferase reporter out of this. So this was really, really quite cool. So just about finishing up here, next slide, please. We can see a bit of an overview of where things are for circular RNA. We have one group working in the clinical phase right now, which is RiboX, and a lot of other groups still in early or preclinical phase stage. I don't think that this is outdated, but this is at least a little bit old now, so I think that this is still current. Next slide. Oops. The themes that we're seeing coming out in circular RNA production are just highlighted here. So in the synthesis and design, we are really seeing the criticality of a good iris or this new innovative 5' cap initiation strategy. And this to me is really exciting because there's this cell or state-specific expression that we can get out of this. For those familiar, everyone in the RNA field has kind of been looking at the LNP field and saying, hey, you guys figure out delivery to make sure we get in the right cells. A lot of people in the LNP field are starting to say, hey, RNA guys, figure out how we can make sure the RNA expresses in just a singular type of cell. So this is exciting because this might be a pathway to success there. There's a lot of ongoing intron development as well for improved efficacy, scar-free synthesis, and as we saw just recently, purification. There's a lot of different purification strategies that we can take here, and it's a bit of a mixed bag on which way you want to take. RNase R seems to be really effective for linear removal, but anyone who's purchased RNase R knows how expensive it is to procure. It's not necessarily the most attractive way of doing anything at scale unless you're preparing it in-house, but just because of the cost, I know it's not an easy thing to do. Bunch of different chromatography approaches. There's this purification approach that we've been working on in our facility here for double-stranded RNA removal. It's not the most scalable because it is a precipitation base, but I'm hoping that we can take some lessons and maybe try to find a way to scale it up. We have a phosphatase, which is used primarily just for silencing those 5' phosphates. And for analysis and functionality, there's a lot of work going on to finding really what the appropriate purity is for functionality, because up until fairly recently, the immunogenicity of circular RNA has not been very well understood. There's been even some papers kind of publishing, trying to review what's out there because it's quite conflicted. And part of this is because there's a lack of standardized guidelines and controls. Next slide. And this is one of the things that we are trying to tackle in our formulation core, RNA and formulation core here at UBC, is trying to begin establishing standards and guidelines for RNA research, because so much of what's out there is not comparable and it's really hard to reproduce. And a lot of this is because there's not really good standards or guidelines for a lot of this. So our funding really comes partly from UBC, but also academic and industry partners. So if you're ever interested in tackling a project together, let us know. And there's our website and my email down below if you want to get a hold of us. And looking forward to the questions afterwards. Thanks so much.
I guess it's my turn. Hello, everyone. It's my pleasure to talk to you today. So my talk centers around continuous production. So this was briefly mentioned by Andy towards the end of his talk. And I guess I'll focus on two key aspects of continuous manufacturing. One of them is around real-time monitoring of the process, because in a continuous process, you would want to see in real time what's happening. And another aspect is how we can reduce costs. And for this, we developed a method that involves recycling of raw materials, which are high cost, as you know, for RNA production. So I guess those are the two key topics. And if you go to the next slide, please. I guess I'll briefly introduce our work at a higher level. So on the left part of the slide, you'll see some of the key components that we are working on. So we do process development. We work on developing continuous processes. We're looking at intensifying these processes. And then we also do process modeling for two reasons. One of them is to use the models to guide process development. So instead of doing experiments, we run the models to test different scenarios, right? And then the models are basically co-developed with the process. There's a cycling between them, right? So it's iterative development of the models and the processes. And eventually, when we have both fully developed models and processes, we can use the models for advanced automation, things like model predictive control. And these can be deployed into soft sensors and digital twins. I'll give some examples later on of the soft sensors that we're developing. And we are doing this work within the frame of the quality by design framework. And we also do technoeconomic analysis. Again, the third component there is this development of new methods for analysis. This centers a lot on mass spectrometry and HPLC. This work is led by Professor Mark Dickman, who is based in the same building here with us at Sheffield. And the fourth component is that by developing dashboard solutions that provide an interface to the process, so that people who run the process can see what's happening there, how the process parameters are evolving, how the product quality is changing, and so on, right? And combining these four together enables us to establish a platform to achieve the aims shown on the right of the slide. So basically, to have multi-product platform capabilities for making products at high quality, low cost, in efficient processes that can enable rapid development and mass manufacturing, and can also simplify technology transfer. If you go to the next slide, please. So in terms of the continuous process, this is how it can look like. So we start from, in our case, we start from IVT, then we do purification, then formulation into lipid nanoparticles, then there's more purification after that. And as was mentioned before by Andy, capping can be done in two ways. We can do analog-based capping or enzymatic capping, and the tail of the poly-A tail can be added two ways. Again, this can be encoded on the template, or it can be added enzymatically as well. I guess you might ask, why are we developing continuous processes if there's already batch processes in place? So continuous processing has several advantages. such as higher productivity. You can run the process at what is called a steady state optimal condition, right? And that just means that it's running more efficiently. Everything is maintained at a consistent level, so quality is more consistent. Because everything is now more productive, it can be implemented into a smaller footprint. And just by the nature of having this continuous processing, which means that you can just run the entire process and have all the equipment basically utilized at all times. And you can have a bit of flexibility in terms of how much you want to produce just by running the process for shorter or longer time periods, right? And because everything is already interconnected, then you can make it into a closed system process, so that can go into a lower grade clean room in a GMP environment. And it can also yield improved quality and lower dsRNA amounts depending on how you run the process. On the other hand, as you see on the right part of the slide, there are key challenges with continuous manufacturing. One of them is that you would want to have monitoring of the process and the product quality in real time. That's not always easy for several quality attributes. And there's generally a lack of standardization and interconnectivity between the different equipments. So we are addressing these challenges. I think I'll talk mostly about the real-time analysis challenge in this talk. So if you go to the next slide, please. In a continuous process, Ashley, next slide, please. Yeah, so as I mentioned, in a continuous process, you would want to monitor the product and the process in real time. And this is currently not possible for several quality attributes. So we are looking at ways of monitoring the process and developing new methods for analysis or for monitoring the process. And if you click, there's going to be a few animations here. So if you click on the slide, Ashley, yeah. So what we know from the IVT, that when you incorporate each NTP, if you click, Ashley, please, then what happens is when you incorporate each NTP, a proton is being released together with a pyrophosphate. The proton comes from the hydroxyl group on the ribose. That's the 3' hydroxyl group. And if this proton is released, and there's quite a few NTPs being consumed, and there's a one-to-one stoichiometry between NTPs and protons, we can imagine that quite a lot of protons are being released, and that could result in a pH drop. And we know that the buffer in an IVT is something like a tris buffer or a Hepis buffer, normally at something like 40 millimolar, so it's not very strong and probably would not hold the pH in place. If you click again, Ashley, then maybe next click as well, then we can imagine that what you can have, if you measure the pH, you would register a drop in the pH during the IVT, and that's actually what we see here. So we started the IVT, and then at some point we added the template DNA, and then actually that's when the reaction started. And as you can see, after the addition of the template DNA, we can see a drop in pH during the IVT, and that actually corresponds nicely to the consumption of the NTPs there, right? And actually, that nicely correlates as well to the RNA accumulation or RNA yield, right? So basically, this shows that there's a direct correlation between pH, NTP consumption, and then there's an inverse correlation of that to the RNA yield. And what we already had in place, we had kinetic models of the process in which each of the species is accounted for. So by using this model and by measuring pH, we can determine the number of protons released. From there, we can deduct or calculate easily the number of NTPs being consumed. And from there, we can calculate the amount of RNA being generated. So if you click again, please, Ashley, then what we managed to do, we managed to set up this one as what we call a soft sensor, so a software-based sensor, which measures pH and uses the model to calculate yield and NTP consumption. If you click again, Ashley, please. Yeah, so that would be a soft sensor. And I guess if you click again, it's going to move on to the next slide. So this just shows the idea of this, how this works. So again, we measure pH, right? and then we feed that pH measurement into two different models. So we have a full-on mechanistic model, which calculates the mass balances, and it uses basically differential equations to do it over time, and it's also predictive, so it can forecast in the future a bit as well. So that's one of the models. That's a mechanistic model, and the other one is this. It's more of a simplified model that correlates pH and pKa and the concentration of the conjugate base and conjugate acid based on the Henderson-Hasselbalch equation. But basically, you measure pH, you feed it into these two models, and you can calculate yield, NTP consumption, and a bunch of different things. And there's a few examples shown in the next slide. So if you go to the next slide, please, then you'll see how this can work. So the red lines there represent the experimental measurement, and the black line and the shaded area shows the model prediction. So as you can see, we can measure pH, and in real time, we can now predict the concentration of the four different NTPs, either individually or combined. And we can also show the, I guess, the standard deviation on that, and also the residuals. If you click on the next slide, go to the next slide, please, then we can also see that we can also measure RNA yield. Based on this pH measurement, we can see how far we are from completing the reaction. And then we can basically track different components there, such as orthophosphates or pyrophosphates. Basically, because we use pyrophosphatase, the pyrophosphate is being degraded. That's why the concentration stays close to zero, right? And we're also looking at quantifying our residuals or the errors in this with this approach. If you click onto the next slide, please. Yeah, so, and this is just the results from the more simpler model, which uses the Henderson-Hasselbalch equation. But generally showing that there's a good correlation between the model predictions and the experimental pH measurements. If you go to the next slide, please, then we're working on implementing this as a control strategy, basically. So we'll have a model, and we'll have an optimizer that optimizes this in real time, and then we can take adjustments in the process. And we are implementing this in an interesting way, where we have a process in which we actually are recycling IVT components back to the IVT. And I'll talk more about that in the following slides. So if you click on the next slide, please. This just gives a bit of a background to that. Basically, we looked at how the cost would compare for a continuous process versus a batch process, right? And we saw that actually the costs are quite comparable. They don't change much when you go from continuous to from batch to continuous. Basically, both the capital cost and operating cost are staying the same, and the raw materials are the main cost driver. And this is looking at the larger scale production. So yeah, obviously, as you might know, you have very expensive raw materials. Capping reagents are expensive, right? Your template DNA enzymes and so on are all expensive. So this just shows that by going from batch to continuous, the costs won't change too much, right? But what's interesting is that the productivity does increase a lot, right? So it can be up to a 20-fold increase in productivity when you go from batch to continuous. And by productivity, we mean how many grams we produce per unit scale, per unit time with the process, right? So we looked at other ways of reducing costs if continuous production alone doesn't save costs. If you could go to the next slide, Ashley, please. Yes, so basically, in a very simplified way, in an IVT, you have NTPs and capping analogs being incorporated into mRNA. If you click, Ashley, then yeah, that's your substrate, NTPs and the cap analogs, and then you have your product is the RNA, and there are different components there that just enable the reaction to happen. These are the template DNA, the enzyme, and other components. And these components that facilitate the reaction are not really consumed, right? And normally what people do, they run this in a batch setup, and after every batch, they would discard these templates and enzymes, which are actually still perfectly functional, but they are just discarded. If you go to the next slide, please, or just click, yeah. And what people do sometimes, they are looking at immobilizing some of these components, right? So there's been work on immobilizing templates or enzymes or both together. But generally, with both elements, normally the capping analogs or capping reagents generally are not recycled. If you click again, please, then we thought, is there a better way of reusing these unconsumed raw materials for making more of the same products? If you click again, please, then we developed a method, and this was work done mostly by one of the postdocs in the lab, Aditya Nair. But basically, this work involved optimizing both the IVT and oligo-DT purification in a sense that we made them fully compatible with each other so that we can directly load what comes out of an IVT onto an oligo-DT column. So what this slide shows is how this works basically. But basically, if you click, Ashley, please. Yeah, so in an IVT, you go from NTPs to RNAs. As you see towards the IVT, you have a lot of RNA generated. Ashley, click, please. And then we load this onto an oligo-DT column. If you click again, then you'll see that the RNA binds. And if you click again, you'll see that these components that are not consumed in the IVT, these include the template DNA enzyme and capping reagents, are coming off the column, but because they were not diluted, they're coming off the way they went in, so they can be actually reused directly for the next round of IVT. And the RNA is eluted, obviously. And if you click again, Ashley, then we'll just top up the missing components, the NTPs that are actually consumed. And if you click again, then yeah, this ends up being cycled, right? In the sense that we can now, yeah, run multiple cycles of these where we have the flow through of the oligo-DT column being recycled back into the IVT. We top up with the NTPs that have been consumed, and we cycle this to make more and more RNA. And there's different ways of doing that. And some would also reduce dsRNA for a few reasons. Firstly, because there's actually high salt conditions in the IVT, and that has been shown to reduce dsRNA, but also because we don't allow a lot of contact time between the RNA and the enzyme so that the enzyme doesn't extend the RNA, so you don't have this 3' extension type of dsRNA formation there. If you click on the next slide, I think we'll show some of the results here. So the diagram here on the top left shows this process again with the cycling, right? So we go between IVT and oligo-DT and we top up the NTPs, right? And then we can see the yields here, right? The yields are measured before and after oligo-DT purification. And this was run for 5 cycles, so 5 rounds of recycling basically. As you can see, there's a bit of drop in the yields, but it's just not losses in the chromatography step basically. And the integrity actually went up slightly. The integrity was measured by capillary gel electrophoresis here, but generally fairly high integrity. If you click on the next slide, please. We optimized this. Well, we developed 2 different buffers. Initially, it was a slower buffer, and then we made it into a faster buffer. So then the process can take, instead of 12 hours, only 6 hours to run these 5 cycles, right? And still having high yields. If you click on the next slide, please, then we'll see actually interestingly that we not only managed to reduce costs with this process, but we can also reduce dsRNA. So this is the dsRNA in the 5 cycles relative to standard IVT condition. There's a 3-fold drop in the dsRNA amount there, and that's based on a mass percentage. And the capping efficiency slightly drops. We're still looking at improving that, but it's still probably acceptable over the five cycles. If you click on the next slide, please. This is the poly-A tail distribution, similar to an mRNA control, as you would expect, based on just a standard IVT. There's no interference in terms of poly-A tail integrity. Again, this was measured by mass spectrometry, and the same was with capping as well. If you click on the next slide, please. Yeah, so basically, we are also now offering services for RNA manufacturing and analysis for several quality attributes. As you can see here, these are the expected quality attributes for our RNA that we manufacture, and these are the methods that we use for analysis. On the right column are the analysis methods. If you click on the next slide, please, Ashley, then I guess, yeah, well, this is my conclusion slide. So basically, for the soft sensor work, we have developed this method that involves measuring pH and feeding that pH measurement into models to calculate NTP consumption, RNA yield, and many other things in real time. So that's quite powerful. And that enables us to monitor the IVT in real time. And yeah, it's a fairly simple way to go from a pH measurement, something which is easy to measure, into getting a lot of information out using models, right? And then we looked at continuous versus batch production, and we saw that both the capital cost and the operating cost are fairly similar between these two modes of operation, but the productivity can increase substantially when we go from batch to continuous. And in terms of cost reduction with this novel recycling method, we saw that we can actually recycle these materials, right, by optimizing both oligo DT and IVT together to work hand in hand. We did several rounds of recycling with high integrity, with the expected poly-A tail length and distribution. Those were measured by mass spectrometry for the poly-A tail length and distribution. The capping efficiency was slightly dropping in this recycling process. So we are looking at improving that. And the dsRNA was actually much lower in the recycling process compared to batch control. And the dsRNA was measured by the Lumit kit from Promega. And I think the next slide is my last slide, where I'm just acknowledging everyone who worked on this, right? So there's a large team behind this, and I also thank our funders. And I guess with that, that's the end of my talk, and we can take questions. Thank you. Khalid, you're on mute if you're talking.
Thank you. All right. Is that better? All right.
Yes, now I can
Yeah.
hear you. Yes.
Thanks, Andy, Andrew, and Zoltan for a great presentation. Much appreciated. We have a few questions to go through, and I know we're ran a little bit over, so we have about 10, 12,
8
15 minutes.
Yeah, 12.
12 minutes. So let's start with that. Andy, this goes to you. What are the biggest challenges for sRNA characterization other than length that may not be applicable for linear or circular?
Yeah, so the biggest challenge is to make sure the regulatory authority understand that this is a different mRNA technology, that it has a different mechanism of action to linear, unmodified or base modified RNA. It is longer, and the specifications are going to be different. That's the biggest challenge we faced, and I think we've certainly achieved harmony with many of the global regulators on that fact, that it's just a different technology, and therefore requires, I mean, very similar assays, but a different approach, because you can't use, it's not a small molecule. You could essentially think of linear RNA more as a small molecule, and from a regulatory strategy, that's what people have done. This is more like a biologic. It's much larger, harder to characterize. And so we've been upfront with the regulators saying, don't look at it as a linear. This is different. This is why. This is how it's working. It has pluses and minuses. And so a lot more emphasis on cell-based characterization. Is it replicating? Is it expressing the encoded protein?
Okay. Zoltan, this goes to you. Has your continuous production been tried with long self-amplified RNA?
We are currently working towards that. We have not fully developed that yet. But yeah, there's certainly a challenge, as Andy was mentioning. There's challenges at every step with that, right? In the IVT purification, analysis, formulation, right? Certainly more difficult to work with than shorter ones.
But, to build on that, Khaled, the continuous flow, which is the intermediate step between, has been tested. So, Nature's Toolbox have used self-amplifying RNA, Centillion have too, and shown that flow chemistry is adaptable.
Okay. This question to all three of you. This is more on terms of the mechanism. Why do self-amplified RNA requires AU? And when you do enzymatic capping on that, do you actually use, get a cap one, or you actually get an AU start? So whoever want to take that one, I would like to hear from everyone on that one.
So the authentic 5' end of an alpha virus is an AU. So,
Okay.
you have you're using T7 polymerase to make the RNA, but in order to, there's also another, it's this tale of two enzymes. You have an RNA-dependent polymerase from the alpha virus that needs to recognize the 5' end, and it wants to see an AU start. And so, and certainly in terms of the cap structure, if you look in the literature, alpha virus is a cap 0, but everyone's using cap 1 because of trying to down modulate immune recognition of that 5' end, and it seems to work just fine. It's not authentic to the genome of the alpha virus.
Yeah, simply put, though, I mean, your self-amplifying RNA, a lot of them will work without an AU, right? You can get them to work. We've seen it with GG. I think I've seen some groups using CleanCap AG type approach, and they will work. But like as Andy said, the canonical is AU, so it should theoretically work best. I haven't actually seen data comparing them directly. I don't know. Maybe you guys have it.
Nothing in the public domain, Andrew.
Yeah. The other thing to mention, though, is there's a lot of viruses, and this is probably getting maybe a little outside of the scope, but a lot of viruses are very particular about those, those first nucleotides, their identity, and also making sure that, like, I think it's the rule of 3 with some viruses, they require an exact number of, like a multiple of 3 for their entire genome for it to be effective. So I'll just leave it at that.
Zoltan, do you have anything to add regarding that?
I guess a lot has been said already, right? Maybe what to add, in our experience, if you do an IVT with CleanCap AU and you don't add the CleanCap, well, it's an AU start, right, on the template. And if you don't add CleanCap AU in the IVT, it just won't start, right? If you have AG, it actually will start without the CleanCap AG in the IVT, but if it's designed with CleanCap AU, you have to have the CleanCap AU there, otherwise it just won't initiate, interestingly, right? So that's an interesting thing that we noticed, and we took a while to figure out why the IVT doesn't work when it's an AU start. Not sure if you have seen this as well.
So a follow-up, can Cap0 structure still work with the saRNA for its replication in the cell?
Yeah, absolutely. Works just fine.
And in terms of if you decide not to do co-transcriptional with AU, and you decide to add your cap enzymatically after post-transcription, does it matter if it's 0, 1 AG, N-methyl, M6? Does it matter?
Are we talking for mice or humans, Khaled?
For eventually it's going to be for a human. I mean, I
don't think from what I've reviewed in the literature, to try and dissect apart what people taken forward, I think it's been Cap 1 that's gone into humans because of the assumption that Cap 0 will be recognized as a foreign. It's what most viruses have as their capping structure, and so it will be recognized. And so from what I can see, most people go forward with cap 1, but I don't think there's been a direct comparison between cap 0 and cap 1 in humans to determine if it's essential.
Okay. So, Andy, you showed some data regarding circular RNA with uridine and N-methyl pseudouridine. You know, it was done in a certain process. Is there any translation data in Cell regarding those two circular mRNA with uridine and N-methyl pseudouridine?
I'm not going to say there isn't any out there, but I haven't seen any that's been functional. As a general rule, those will just modify the iris structure too much for there to be efficacy.
Okay.
But I think that there is some potential to engineer irises to incorporate them. It may be possible in the future.
So on that, you know, most of the work that was done on polymerase, especially mutant T7, is with the goal is to reduce double-stranded, but mainly all the data is with N-methyl pseudo-U. But we know with circular you need uridine, not N-methyl pseudo-U right now to do that. Has any of those worked, or do you feel we need a new enzyme that is not necessarily mutated T7 to truly reduce double-stranded, so that way it could help the manufacturing of circular RNA?
So particularly towards circular RNA, I think there's potential there. We actually are evaluating lots of different T7s. We have actually, just by happenstance, evaluated with saRNA so far. We haven't started the circular RNA analysis. And I think there's some potential there, but I think a lot of the challenge with the double-strand RNA and circular RNA comes from the fact that there's those introns, and I don't have the data to prove this at all, but I suspect that those are acting as primers for RNA extension to create more double-stranded RNA all over the place. So just because of the extent of double-strand RNA that we see in circular RNA, that's my hunch at the moment. And I think it's going to be really hard to get an enzyme that just won't use things as primers if they're available. And that's where maybe continuous manufacturing can really play a really strong role, because the idea is we want to try to remove those impurities as soon as possible. And the other thing with continuous manufacturing that I think has a lot of potential for circular RNA is you want to try to separate circular RNAs as far away from each other in the reaction as possible, right? Practically speaking, there's kind of a lower limit of what you want to do in a reaction. But if you can separate out circular RNA as soon as it's made, then it has much more likelihood of circularizing on itself rather than finding neighbors and having other weird things happening.
Okay. Well, we're up on time, so I really want to thank our speakers again and all the attendees. Thank you so much for joining us today. That was great. And you will, check the AMM website. You will find most of these webinars recording, that if you want to visit them later, or passing them on to your friends to learn more about mRNA. Thank you so much, and have a great day or evening, depending where you are in the world. So
Thanks so much.
Thank you, everyone.
Thanks, everyone.
thank you, everyone.